I hope everyone's first semester is off to a good start. I'm Robert McDougal, and I run a dry lab that mostly focuses on applying informatics techniques, broadly defined, to seek insight into neuroscience, although we're starting to diversify as we see opportunities to apply our tools to other domains. To give you a sense of my background, I started out as a math PhD, and I did a brief postdoc in computer science before migrating to the med school to do computational neuroscience work here at Yale, actually, in the former neurobiology department. This led to a postdoc at the Center for Medical Informatics, where I got my first formal training in ontologies, NLP, machine learning, modern databases. I brought these techniques back with me to neuroscience as a research faculty for a few years before joining biostatistics as an assistant professor in the health informatics division. So in addition to CB&B, I'm very actively involved in the health informatics master's program. I teach CBB 634, which is computational methods for informatics, where we discover the math and logic that make informatics research possible. With my neuroscience background, it's perhaps unsurprising that that's a key focus in my work. However, it's just hugely important in general, right? There's about 1 billion people worldwide that have some form of neurological condition affecting their quality of their life. I mean, so 12% of the population has migraines. That's huge impact on people. If you think I'm talking about dementia, there was a 2017 estimate that dementia alone was projected to cost the world economy over $1 trillion. And that's just going to increase as the population ages. So this is a huge public health issue. And so while we do have this clear biological focus, our work itself is fundamentally informatics, and that takes several forms. So we work to improve the quality of simulation studies through work with standards boards and scientific infrastructure projects. That part of our work is more technical than scientific, and I debated whether or not I should mention it at all. But really, that kind of work, this infrastructure work, does help make science and other people's science better, and it does lay a good foundation for informatics. But we are ourselves very interested in developing new computational techniques for extracting information from scientific artifacts. So there's lots of people here at Yale, and we're ones of them, that'll do text mining and NLP stuff. And we're definitely interested in that. But we're also interested not just in looking at the scientific literature, but there are other things that scientists create. So we're also interested in what can we learn from scientific codes? If you do simulation, can we infer information out of that? We apply our simulation techniques and our literature mining to gain insight into biological processes. And this includes, but not limited to, the things that we talked about. So working memory, ischemic stroke, and Alzheimer's. One of the exciting challenges with these problems for me as a mathematician is that the dynamics happen across a range of spatial and temporal scales. And you really can't get a full picture of what's going on unless you figure out how to study the interactions of these scales. So just very briefly on the scientific infrastructure side, so there's more that I'm not going to talk about, like standards work, but our two most widely used resources are contributions to the Neuron Simulator. So we have one of 2 grants that's primarily about developing this tool. In our case, we're focusing on chemical signaling within neurons and across the extracellular space, as well as how that interacts with ion channels that drive action potentials. And the ModelDB repository, which is a discovery tool for over 1,600 published, curated computational neuroscience model codes. So we provide various visualization and analysis tools. But equally important, this collection of models also serves as a data source in its own right, as each model can be thought of as a miniature review of a specific area of the scientific literature, right? If you're making a simulation, then you had to make a decision, for example, about what cell types, what ion channels, what morphology, what is necessary to represent your area of interest. And so collectively, these models can be used to make inferences about what's known as about what's known about the biology, which brings us to the science. So I'm just going to give you a taste of some of the things that we're doing, but I'm happy to talk more about these or other projects privately later. So we're very interested in learning about the structure of neuroscience research so that we can better identify novelty, identify correlations, and sort of build upon what we've started to do, which is to try and build a very, at the moment, rudimentary, but knowledge graph of the field so we can make inferences and comparisons. in an automated way. So most of this is hard, but there are some things that are easy. And just to give a shout out to this, if I look at it within our database, a collection of these models, and I look within this, what has been studied when we look at Parkinson's models? Well, what falls out? Well, okay, well, the most common thing that's studied in a thing that's about Parkinson's is deep brain stimulation. If you know anything about Parkinson's, this is not surprising. It's basically, it's one of the most prominent treatments that's available for Parkinson's. But it does sort of just fall out of looking for correlations. And so we can make other inferences about cell types, brain regions, and things like that. We can also, again, pretty readily, if we start including in our metadata what the literature citation is, we can look at this collection of, well, what do we know about Parkinson's? And then, again, so here what I'm showing you is strictly from the modeling literature. But what lays the framework for understanding of how we would represent Parkinson's in terms of equations, how we would represent Parkinson's in terms of computers? And so this is a joint thing, right? We need to know something about the biology. And indeed, thankfully, the highest cited paper in our Parkinson's collection is a biological paper. It's an old biological paper, but it still lays the framework for understanding of, hey, look, Parkinson's is basal ganglia disorder. What's going on? What's driving that? What's the architecture? What's the anatomy? But then we can also identify key modeling papers, like this work by Dave Terman, who, shout out, was my dissertation advisor, and some various other papers that sort of lay the framework for how we might go about studying Parkinson's and simulating Parkinson's, and what do we need to know. So we can dive into this authorship network even further to make inferences about the authors, and that'll help us understand how to interpret papers by authors. So we find a new paper by a given author. Is it likely to be a simulation study? Is it not likely to be a simulation study? Is it somebody who really knows about the biology? What is the role? What is this author bringing to the paper? How can we do this? Well, since we have this authorship network within the subdomain of computational neuroscience, we can look at all the papers that cite each other, other papers, and then we can make assumptions. So it turns out if you look at the centrality, which is a measure of connectedness of the network, authors with high centrality tend to be primarily simulation focused, which makes sense in a network about simulation papers. So you can distinguish for a given paper, oh, hey, look, it looks like Michele Maiori probably did the simulation work and he collaborated with these people who, if I look at if the same graph in the Parkinson's literature, I can say, yeah, they're clearly influential people on the biology side. So this allows us to make sort of inferences here. And while I'm showing everything that I'm showing for this is going to be computational neuroscience focused, I just want to also say that we're starting to turn these tools specifically towards Alzheimer's in general and COVID-19 as well. So we look at using various parts of papers and their utility for obtaining different types of information. So here, for example, we see that we can classify, we see that we can use even just straight up the paper titles to distinguish general papers from PubMed, these purple papers, from neuroscience themed papers, these yellow and green. And PubMed is generally separable from neuroscience related things, although there's certainly some purple dots over here. There is, however, less separation between, in the title, is this a paper that involves a simulation work, or is it a pure experimental paper? As a CB&B professor, I would encourage, I'm actually a little disappointed by this, because I would like to believe that regardless, simulation is just a tool. You build a computational model, you build an animal model, either way, it's just a modeling tool. You're trying to study a biological question. I would like to believe that in the ideal, that it wouldn't be separable on the paper. It would be, what's the biological question? What are we focusing on? But statistically, that's not true. We are actually able to make inferences about what is or is not computational based on the title, although at least it's a little fuzzy. Perhaps unsurprisingly, if you look at the abstract, we get a little better separation. Just very briefly on this. So you can look at this because there's a lot more information there, right? And you can look at all the words in the abstract. And so, right, all other things being equal, if I have an abstract that includes the word simulation, well, that's going to increase the likelihood that there's a simulation study as part of how they studied whatever. Likewise, if the abstract includes the word experimental, this also increases the likelihood that it's a simulation study. My guess on this is basically that if you're doing experimental work, you don't feel the need to qualify it as experimental work, whereas simulation people do. So we've taken this and we've extended this to different types of data within these things. So if I want to know about ion channels in a simulation study, well, you're probably not going to mention that in an abstract, because if you're really trying to study Parkinson's, unless you're saying, oh, well, this ion channel is upregulated or downregulated, it's not going to be worthy to be mentioned in the abstract, but it is part of understanding what the simulation is doing, right? So that you can look at the whole paper and you're not going to be too led astray. If on the other hand, you're looking at brain regions, important brain regions tend to get mentioned in the abstract. And if you look at anything else and you find stuff that's not in the abstract in the rest of the paper, it's probably not actually what the paper is about. In terms of simulations, so I'll be very brief. Biologically, our interests are generally in regions where the cell and systems biology interacts with classic neuroscience concepts like networks and ion channels. So models generally include Hodgkin-Huxley style ion channel kinetics, spatially extended neurons. Here, for example, we're looking at a wave of elevated extracellular potassium that spreads over a 2 millimeter by 2 millimeter slice of the brain. When we do these studies, we're looking at every neuron is simulated individually. And the reason being that this does let us see sort of the statistical variation. So it's not a perfectly round wave that spreads. There are some fluctuations in the boundary that may affect the wave speed and possibly ultimately survival. And if you're talking about ischemia and the penumbra. So on a different scale, we've looked at calcium-induced calcium release in the ER. And this is interesting because it replicates some of the phenomenon that you see on an action potential, but inside the cell and in a slower level. I'll leave the science at this so that I can just give a shout out and say that if you're interested in anything that we've talked about, feel free to consider doing a rotation with us. I mentioned these projects in the BBS rotation guide, but rotations can be an opportunity for everyone, both students and faculty to some extent, to explore. And if there's an opportunity for a shared interest, let's talk about it. And with that, thank you. And